What's up, APBio penguins and recappers? Tonight is the final countdown, the last Q&A. So don't forget that we are going to go over basically everything. So whatever questions you have, you can go ahead and drop them down into the little chat. I'm going to say enter your questions in the question box. And that way I can get us a... So we can go ahead and get this message pinned so that we will have it pinned for the future. Okay, so I think I'm pinned. All good to go. Melanie should be joining. There she is. Hey, Melanie.
Hello. Here we are. The last
Here we are. The last one. How are you feeling?
I'm good. I am excited. I think my students are ready. I think right now it's all about building the last minute confidence, and that's what we're going to do for you guys here. Right. Build that confidence.
Yeah, so I am Tiffany Jones from APBio Penguins, and you are?
I'm Melanie King from the APsolute RecAP.
Okay, so tonight we just want to go over whatever questions you all have, right? So before we get into it, let's do a really quick, just what are the 8 units, just so we have a general idea, okay? So unit 1 is all about biochem. Okay, so it's all your macromolecules, all your water properties. Melanie, want to do unit 2? I'll let you do.
Yeah, let's go back to unit 2, cell structure and function. And no, the mitochondria is not the powerhouse of the cell. Don't you dare. Circulatory volume ratio, big theme.
Unit 3 is all on cell energetics, so cellular respiration, photosynthesis, and then enzymes.
Unit 4, cell communication and cell cycle.
And then unit 5 is on meiosis plus genetics, so like all that genetic stuff.
Unit 6 is going to be, my goodness,
molecular genetics. So
there we
like
go.
Sorry, my gosh. Replication, transcription, translation, genetic engineering, all of the different biotechnology, all that fun stuff.
Natural selection and ecology, 7 and 8.
Natural selection is 7, and then of course unit 8 is ecology. So if you will go ahead and pop your questions down into the chat. Before
also,
we get into that,
when is everyone taking their exam? Just let us know in the feed. Are you going to be this Friday on the 14th? Or are you going to be one of our digital test takers later on? We will talk about the different administrations at some point today.
That's what I was about to go right into while they were kind of getting their questions popped in. So there are different administrations, whether we're doing paper or whether we're doing digital. The only big difference between the two is that, of course, the date in which the paper one is going to be this Friday. And then the digital ones, there's one on the 27th and then one that's at June 11th.
That sounds right.
Is that right? Okay. And I believe that the June 11th and the May 27th one are at the exact same time everywhere, no matter where you are. So is it noon my time?
I think it's noon Eastern time. Yeah, noon Eastern time.
But other than that, the big difference is that the second question, your second free response question, traditionally you're going to have to graph. Like on the paper exam, you'll do a graph. But on the digital exam, there's going to be some alternative way they're going to ask you that information. So you will not have to graph on the digital. The same thing for question 5. You won't be drawing anything. Usually number 5 has some type of drawing, whether it's a pedigree or whether it's a cladogram or whether it's just draw this pathway in the cell. There won't be a drawing. You're going to assess it some other way.
And for a lot of ways, it's pretty minor differences compared to what some of the other subjects are experiencing. So neither one is harder. Neither one is going to be scaled differently or count more. It's really about how you show your content knowledge. So don't put yourself in like false security that, oh, you're taking the paper test, it's going to be easier. It's just a different strategy of test taking. And that's really where you have to like shift your mindset based on how you're taking it.
So how long are you telling your students about pacing wise if they're doing the digital exam, since they can't go backward? So what is your timing?
Multiple choice, I tell them to aim for a minute because there's going to be those they finish in less than a minute and those that need longer. You know, on the paper pacing, it's a lot more like 90 seconds. But then again, you have your set questions and your non-set questions. So you want to even it out. I know it's going to be so hard to ignore the clock, right? It's going to be there. But, you know, check into it periodically. Like if you physically have to put a sticky note over it that you only lift like every 15 minutes or every 10 questions, like maybe that's your strategy. but it's only your strategy if you've practiced the strategy.
Right.
Don't fall into that.
Right. And so someone asked real quick about cameras. No, you are not required to have a camera for the digital exam. And you're also not supposed to have any type of notes. Okay. So they've specifically said no notes. They will not help you in any way, shape, or form. The questions have been written so that they won't do anything for you. So Melanie, you think we're ready to get started with our Q&A?
Yeah, we're going to be quick, guys, just so you know. This is going to be a brief dive into the topics that you guys put in Q&A. And if something pops up in the meantime, just add it to the box and we'll get to as many as we can.
So let's just do real quick. What is transcription translation and what's happening in those processes?
So central dogma, big picture, right? DNA into RNA into proteins. So location matters, and the type of cell that you are matters as well. So transcription and translation are going to happen simultaneously in prokaryotes practically because there's no nucleus, right? You can't divide it out. So you're also going to have less like processing of your mRNA transcript, actually none, because it overlaps. And then when we're in eukaryotes, right? So we're going to transcribe our DNA into RNA, and then we will modify our mRNA transcript with, getting rid of our introns, our poly A tail, our MG-cap, and then go out to ribosome for translation using our codon chart, which you do not have to memorize, but you have to know how to use.
And traditionally on the exam, that codon chart is given in like an actual chart form. So some of your teachers may have done this like circle thing. It's usually a chart. It's actually since I've been looking at the exams back in like 2013 and on, it's always the exact same chart. So if you've seen one of those charts in a question that your teacher has given you, you should be prepared for what that chart looks like. So Melanie, are there anything like that violates the rule in terms of your central dogma of going DNA to RNA to polypeptides? Is there anything that violates that? Like what kinds of things violate that?
What are you talking about?
I'm talking about retroviruses.
You're trying to tee me. Oh, retroviruses.
No, I'm talking about retroviruses because retroviruses are one of the viruses that's actually in the curriculum. And they have to make sure they understand that the RNA is the genome. And then they're able to go through reverse transcriptase to make DNA. And then from that DNA, it goes through DNA to RNA to polypeptide. So I want to make sure I mention that since we were talking about transcription translation.
Yeah, we have
I wasn't trying to trip you, I promise.
No, it's funny because like these small little moments, like they'll leave your brain, then they'll pop back in. But just remember, if you're transcribing, you're still in the same language. So you're still in nucleic acids. It might be RNA, it might be DNA, but you're still in the same molecular language. When you translate, that's going to put you into a different chemical language, amino acids, and its primary structure is what you're going to form.
Okay. So now let's jump over into mitosis versus meiosis, right? So there are these certain different names that we give these different molecules as we're going through these processes. So let's first think about
they all begin
mitosis.
with CHR.
I know. Mitosis to meiosis, right? So in mitosis, we're trying to make more of our somatic cells or our body cells. Versus in meiosis, we're going to make more of our reproductive cells, okay? And that's where the big reasons why we see the differences that are happening, okay? Because our mitosis, we're making more diploid cells. Meiosis, I'm making haploid cells. Okay, in order to start the same exact way and end up in a haploid cell, I have to go through 2 rounds of division with meiosis versus 1 round of division with mitosis. Okay, so another thing is what is being divided in these processes. Okay, mitosis is going to be your sister chromatids. Okay, so sister chromatids are genetically identical. Okay, you have your chromatid, and then you have its complete identical duplicate, which is its sister. Okay, and actually together, they're still called chromosomes. Okay, so be very careful on your wording there. They are sister chromatids or also chromosomes. Okay, when we talk about meiosis and I have my tetrad, I've got my homologous pair. So I've got the sister chromatid plus the chromatid of my maternal, sister chromatid, the chromatid of paternal. This together makes homologous chromosomes. So these would all be, of course, the same height, but my fingers don't do that. They would be the same height, same length. They would have the same in terms of loci of different things that they code for, the same, but they could be different alleles.
And so you talked about chromatids being identical in mitosis. In meiosis, why sometimes our sister chromatids no longer identical as they
during
through.
prophase 1, they go through a process called crossing over. So the 2 internal parts, so these are non-sister chromatids, they're going to overlap. And when they overlap, they can exchange their genetic material, which allows these 2 interior ones to be recombinant chromosomes versus these ones on the outside are considered parental chromosomes. So during meiosis 1, we're going to split our homologous pairs. Okay, so this is where we see this split happen. Okay, and then during meiosis 2, that's when we end up splitting the sister chromatids and sister chromatids. Okay, so there's a big difference there. So I think I had answered
meiosis 1, you're going to go diploid to haploid, and then in meiosis 2, you're just going to split sister chromatids, so you go haploid to haploid. So be mindful of that. So if you're looking for something familiar to you that is like the X structure, they're still connected, that has sister chromatids still attached at the centromere, and then they can pull apart. So we pull apart sister chromatids in meiosis and mitosis. It's just at different points.
Okay, so negative versus positive feedback. You want to take it?
Sure. It's kind of intrinsic to the sentence. So negative feedback is going to turn something off. Positive feedback is going to cause something to happen. So I like to think about like thermostat in your house, right? So set it at 70 degrees. The house temperature warms, system kicks on. When it goes back to the set point, it shuts off. Right. And so that's not going to be the case in terms of a positive feedback mechanism. Negative feedback is more common. And what we see, a good example of negative feedback inside the body would be your blood glucose levels and your insulin. This is an application question. You do not have to know body systems on the exam. I think that was a question we had later on.
Yeah. So there may be body systems in questions that you see. So they may be talking about the immune system, or they may be talking about the nervous system, but in actuality, those systems you don't have to know, but they'll be application questions. So they'll give you everything you need to know in the question. There's a question that I do want to help a student with, but I need to pull something up. Yeah, can I throw a cell organelle question to you and you answer it while I get this pulled up for the other student? Okay.
Let's hear it.
So the student wants to know, I forgot I'm supposed to be asked the answer, like
I'll read them out. It's fine.
Yes.
How does the Golgi apparatus, RepER and SmoothER relate to each other? Okay, welcome to the endomembrane system, except, okay, you included them all. So endomembrane, meaning they all have phospholipids, they're interchangeable, they connect through vesicles. So if we think about their roles and their functions and how they all have a large amount of surface area, we are going to produce a protein in the RepER. It's going to be vesicled off and go towards the Golgi apparatus for packaging and tagging, perhaps for an intracellular, extracellular transport. The SmoothER is going to be involved in lipid synthesis. You don't have to know a lot of specifics about the SmoothER, but they are all interconnected and they all connect to the nuclear membrane on diagrams. So just because it has membranes doesn't mean it's going to be part of the endomembrane system. The mitochondria, the chloroplast, not part of the endomembrane system, but those are going to be organelles of endosymbiosis. Just like a heads up about organelles. You've learned their functions in your first year biology course. So you're not going to be asked, what does the RepER do? Okay. And when you are explaining these structures, you know, it's not, just try to elevate your language a little bit, right? Analogies, they're great for like minimal understanding. Analogies are not answers. So if you find yourself wandering into analogy land, like try to pull yourself back and go towards very specific application-based, back to the prompt, you know, concepts for organelles.
Thank you. I couldn't find the question that I need. I can't find the thing I needed, so I'll look back at that in just a moment. Like we're gonna do something else, then I'll go back.
Okay.
And I'll look again. So I'll answer this question for them. So our next question is on our Operons. Right?
You're it.
I know, that's fine. I'm just gonna take this one. So in terms of, there's two different types of Operons, right? And so there's an inducible Operon, which if you've ever kind of heard adults talking, they talk about someone gets induced into labor, like they're trying to start labor. So an inducible Operon is something that's turned off and gets turned on. So we turn something on somehow, right? And so if we're needing to turn it on, it should only be on in the presence of the thing that it needs, right? To need breakdown. So an inducible Operon is going to break something down. So we talk about the lac Operon. So if there's no lactose present, I don't need to have this sequence of DNA on to make a protein to break down lactose. Versus a repressible Operon would be like your trip Operon. Your trip Operon is going to synthesize tryptophan. So if there is no tryptophan present, then I'm going to go through this process to make it. But if it's there, it's going to repress it, negative feedback, to stop the pathway. Okay. And so in terms of these Operons, what they'll do is they'll give you a picture of the Operon. Okay. And then they're going to show you different materials that could be bonded to it. Okay. If your repressor is bound to your operator that sits inside of your promoter. So let me first say what these words are, right? The operator is going to be the location where the repressor binds. The promoter is the location where RNA polymerase binds. So if the repressor is bound to the operator, it blocks RNA polymerase from binding to it blocks RNA polymerase from binding to the promoter, so the whole thing is off. I just lost Melanie. Oh, bye, Melanie. We'll see you in a little bit. And then in terms of our lac operon, the inducer is going to bind to the repressor, turn the repressor off. So then we don't have anything bound to the operator, and then we'll be able to see the RNA polymerase can then bind again. And I'm only laughing about the fact that Melanie left me because last year at this exact time, and in fact 5 days from now, in the past, her and I did a session in which she dropped out of the session so many times that I ended up taking the session myself. And so I think we tried like 3 times to do our big Q&A session. So that's why I host.
To be fair,
you've not dropped yet this year.
This one was user error. I was scrolling through the feed and then I scrolled too hard and closed Instagram. And all of a sudden I was gone. I was like, user error on that one, not internet error. That was all me. Sorry, guys. Hope you learned a lot about the lac operon in my absence.
Did great with the lac operon. You want to take a cellular respiration?
I suppose. Let's see what we got.
It's up to you. I can throw it at you.
This is a glycolysis question. I heard that earlier.
There was a glycolysis one in there too, but this was a Krebs cycle one. And I'm going to go back to trying to find the picture that the student needs.
All right. So when we come down to cellular respiration, and I'll just talk photosynthesis briefly as well. Location of process, what goes in, what comes out, and kind of like why. Like, what are those components for? Don't get hung up in the numbers of things produced necessarily. I know that's an unpopular opinion. And all of the major steps. Like, if they want you to interact with the Krebs cycle in some way, it's actually pretty probable they'll give you a diagram of some variety to interact with. That's true for the ETC
they did.
as well.
2014 question that they had everything for you.
Yeah, and so it's kind of like a nice, no, that's fine. I mean, it's kind of like a nice little like alleviation point. So, like Krebs cycle, right? So we are in the mitochondrial matrix, the fluid component of the inner, inside the mitochondria. This, as a point of analogy, the Calvin cycle is in the stroma. So that's the fluid portion of the chloroplast. You kind of have this crossover here. If we think about the what goes into the Krebs cycle and why, right? We are forming our ATP, right? That's going to be the goal here. So we want to pull apart our oh my goodness, I'm totally like speaking backwards here. We want to pull apart our sugars. So we have, to this point, we've gone in, we started with glucose down to 2 pieces of pyruvate, then we pull off another carbon down to acetyl-CoA. We're finally going to finish pulling all of those carbons off. And in the process of that, we are going to create some ATP and our electron carriers of NADH, which are going to go to the electron transport chain at the end, right? So we finish pulling apart our carbons.
Okay, I found the question that I wanted to do for the child.
Awesome, okay. Awesome.
Because I think that it's something that a lot of students might need. So now I have to actually find the question in the chat again.
Oh, goodness.
So which I probably don't have to, but
hey, Brian,
I'd like to try to
put the question in the question box so we don't lose it in the feed.
Okay,
we will talk about autosomal and sex link, absolutely.
The person asked, and I can't find it now, but it's about how do you do a cladogram? Like, how do you fill in a cladogram? Okay. So we're doing our cladograms, and that would be an example of what you could see maybe on your question 5, right? Where they're going to give you the generic cladogram structure, the template, and then you're going to have to fill it in. Okay, so I'm going to flip my screen. So because I pulled up a question, I pulled up the 2015 question. I think it's like number 5. I scrolled 2 down so on it. Okay, so they'll give you a chart. And on this chart, you're going to try to figure out, okay, well, what's most closely related? You're looking for numbers that are small, okay? The smaller the number, the more closely related they are. So you see right here, there's a number 1, and then here we see a number 3, which means that this one, E. africanus and E. ferus are closely related, and then A, whatever that word is, foresty, and then Gallus gallus, which is actually a chicken, are closely related. And so if you look here, you can see that there's 2 branches. So you would put the E. africanus, E. ferus, and then you would have the Gallus gallus, and then the, A. foresty together, which then puts the one that hasn't been talked about yet was the depolyptus. So
there you go.
Completed. Let me go to, I have it already complete on the screen. So this is what it would look like. And then any of these can actually be rotated. And I am running through this really fast because I know that we have other things that we need to do also. But I've already done this question like 3 different times in my FRQ session. So
there you go.
You can easily watch one of those FRQ sessions, and I've gone over it so that you would have that deep understanding of how to do all that. But I wanted you to at least visibly see how would it show up on the exam and what am I looking for? I'm looking for small numbers. And if there's a small number, there's going to be like a little branch that goes on them. Okay.
I hope that helps. I always start with the out group first. I go the other way.
Do you really?
I do.
Oh, well, that's why we're both here, right? One of us has one way and one of us has the other way.
And yeah, really so if you're looking at a cladogram with a friend and you're comparing answers and they're different, they could both be right? So it's because that pivot happens at the node, and it could happen at the node, just because they're physically adjacent doesn't mean they're closely related. They have to connect back to a node. Imagine like you're driving along a path, and half the table is blank because it would just be redundant information, right? We're comparing one column to one row, and that intersection is where you compare their differences, but the rest of the table is blank because it would just repeat.
Yeah because this should be the same as the mirror image, which is right there, or something rather. It's a mirror image. Oh, so like over here, I think. Sorry. No problem. Okay.
All right. How can I tell in a pedigree whether a trait is autosomal or sex-linked? So autosomal, chromosomes 1 to 22, sex-linked, chromosome pair 23, just so we can have those terms kind of like identified. What's your trick? Like, what do you do first as a student?
I always look for dominant versus recessive, because if you're seeing it in every single generation, it's sometimes most likely going to be a dominant if it's in every single generation. If it's recessive, you'll see it jump generations. And on the exam, they're going to make it obvious. It's not going to be it could be. It's going to be obvious. It may not be obvious on the entire pedigree, but there will be a section of it that it's completely obvious about what it is. So if you see it jumping generations, then you know it's recessive. And then from there, I kind of look for a affected female and an unaffected male. So I kind of follow the female to male because the male gets its X chromosome from the mother. And so that's where I follow it. How do you do it?
No, similar. I always do dominant recessive first. I feel like it's the easiest part to figure out. But the thing that needs to be kind of recognized is that it has to be always true. It's not like, well, it's mostly dominant. Like, that's not a thing. Like, it has to always be true. And if you think you figured it out, throw a genotype on there and follow it through and see if it makes sense. If you get stuck to a point where it can't be possible, then you have the wrong pathway. Now, don't spend too much time on it though. Like, if you really are stuck, give it a guess and move on. Go back to it if you know that's your Achilles heel.
There will be other things you might see in these pedigrees in which, there's an affected female and all of the children are affected. So, any idea what that?
This is a good day for that one, right?
It was a great day for that one, isn't it?
Thank your mothers, everyone. It's happy Mother's Day, Mrs. Jones.
Thank you.
It's your mitochondrial inheritance, guys. So, and ladies, it's going to be inherited through mitochondria. It is not nuclear DNA. So non-nuclear inheritance is sometimes how that's expressed.
And that isn't just in terms of the mitochondria, that also applies to the chloroplast. So there hasn't been a question I've seen yet, but we always focus on the mitochondrial inheritance and we don't really mention the
we never do.
What do we call it? The chloroplastal? Like what do you call it?
Whatever we decide.
I don't know. We'll name it ourselves, okay?
Yeah, I mean, I'm sure it has a name, but yeah,
it probably
while we're
does.
here on chloroplast and mitochondria, guys. I apologize. I teach an all boys school. That's my vernacular keeps coming out. But they have their own DNA, their own ribosomes. They replicate independently because they were likely once their own prokaryotic cells. It's well, I like your strategy for this one. It's holding.
It's holding.
And the P stands for photosynthesis. It doesn't, but that's how I remember it.
Right. So the reason I'm pulling this up is because there's between cell respiration and photosynthesis, there are two words that sound very close together, but it's actually an acronym. And it's NADH and NADPH. Both of these are electron carriers. So that means they're going to hold That's what the H stands for. Hold, I know it's a lie. Hold the electron to then carry it somewhere else. Okay. And then the P that you see in NADPH is going to only occur in photosynthesis. Now, question, Melanie, does cell respiration happen in plants? They have a chloroplast.
Sure,
Do they do cell respiration?
Sure does.
Ooh.
Oh my goodness. They make both, right? So they
Yeah.
make their own sugars and they use their own sugars. They have to do both processes. Otherwise, they would be very inefficient.
Yeah. And also at nighttime, I know that there's the moon, you can do some stuff with moonlight. But for the most part at night, they're going to need to photosynthesize, like they can't photosynthesize. So they're going to have to use their stored sugar as well as the other parts of the plant. The whole plant isn't
Green
leaves, right? It's not all green. So the roots and the trunk and the stem and all the other parts of the plant are going to need to go through cellular respiration. So the only part that goes through photosynthesis is that green leaf part, and it makes enough food for the entire plant. Like, that's just crazy. Plus, some of them make extra food that we all eat in terms of, like, you know, potatoes and stuff. Like,
carrots.
Whoa, carrots.
Yum.
Like, what is that craziness? Sorry.
And since we mentioned plastids, chloroplasts are not the only plastids. I just had to say that out loud.
Right. There's the apoplast and the chromoplast.
Yeah.
There's all those
but they all
types.
are energy transducers. They all just use different pigmentation. Same idea.
Okay, so in terms of gene linkage.
Oh, you mentioned recombinance earlier. Don't be scared by the term. It's just recombined DNA. Ooh, recombined DNA. Have you
Don't close out again.
Gene mapping questions on the exams before?
There has been gene mapping questions before in terms of like what's the recombinant frequency, I think. But like it's not like a super, like they've never like made a map that I like.
Right.
Is there a map?
Gene unit.
I think there may have been a map like back in the day, but I don't think it's gonna be on there now. But what you may have to do is determine in terms of how far apart they are, based on the information you're given. So first thing is thinking about linked versus unlinked. So if something is linked, it means that it's close together on the same chromosome. So it's close enough that it will be inherited together. Okay. So two things can be on the same chromosome, but be so far apart that because of crossover, it appears like they're on different chromosomes and they still go through independent assortment, which was that process that we see in meiosis, in which our chromosomes, our homologous chromosomes line up on the metaphase plate during meiosis 1. So that'd be metaphase 1. Anyway, so due to the crossing over, we could have the recombination take place. So if you're looking at offspring and you see that there's a whole bunch of them that look like the parentals, okay, so I don't know, like 400 parental, 400 parental, and then there's 100 and 100 that are like combinations of the parents. That is probably going to show you that it is a linked trait because of the fact that we have a very small number that are recombinant. But if you see like 250, 250, 250, 250, that's going to say, okay, well, those are unlinked. They're, even if they're on the same chromosome, they appear like they're not. They're far apart.
Yeah.
And then you'll never get a recombination higher than 50%. If you did, you made an error.
Yeah.
So I hope I've answered
so you're looking at quantity. So whenever we talk about frequency, it's parts of a whole, how often is something happening. So you're going to have your answers either usually in percentages for that. And gene mapping and along a chromosome is just to give us like contextual distances. They're not physically measured. They are mathematically ratioed distances.
Okay, I'm going to throw this one to you because I know that you did a podcast on this one. But you did a whole podcast.
I do podcasts on lots of topics.
Okay. So anyways, the question is about how does cyclin-dependent kinase regulate cell cycle? Well, a student called me out on this because apparently there's more than one CDK.
Oh, there is.
Apparently, they're all over, like in the entire process. And so it's just like an internal signal. And when you build up to a high enough concentration of cyclin, it will allow that enzyme to be activated. And so that's all that we're looking at with CDK. But the primary one that we look at is the maturation promoting factor, or some people call it the mitosis promoting factor. And this has to do with that once we have a high enough buildup of cyclin through interphase, we finally get to a high enough level that the cyclin-dependent kinase depends on cyclin. So the cyclin binds to the CDK, or cyclin-dependent kinase, which will allow the kinase to phosphorylate so that it can move into the process of mitosis. It's basically to say, yes, all of my DNA has been replicated. There are little to no errors. It's just a way to ensure that we are ready to go through mitosis, because that's one of the things that leads to cancer. If you have errors in your DNA, if you have, you haven't finished replicating it, or even you can think about non-disjunction in a way, right? Because if you go through non-disjunction during mitosis, what phase is that in again? With non-disjunction? Okay, so during anaphase, that would be involved where we're, well, my computer's turned off. That's going to involve where our sister chromosomes do not come apart, or the homologous chromosomes don't come apart, which could lead to an additional amount of chromosomes in one cell or the other of that specific chromosome.
Do you find, so have you seen a question about cyclins and like cells that go into G0 or G0? Has that been, I mean, because in reality,
I haven't seen anything about that.
I don't know the answer, actually.
No. Hey, Miss Kaufman came.
Oh, come on.
She was going to have her Mother's Day celebration, so she knows she's going to get to join us. Does she want to go live with us?
I don't know. When she puts a request in, we'll have to approve it. Depends
we'll
if
have
on
to.
Couch or not.
Okay, so what were we talking about, Cynthia? We got distracted by Kaufman.
That's fine. She's answering a cycling question.
Okay.
Yeah, you mentioned phosphorylation. Phosphorylation we see a lot. It's, again, large word, just means stick a phosphate onto a complex, typically a protein, and it causes a conformational change of some variety. It could be to activate or deactivate if you take the phosphate off. We see this happening a lot.
And a lot of the stuff we just mentioned is all about cell communication, right? Yeah. So different cells are going to go through different pathways, even if they have the same signaling molecule. And it's because of the variety that happens within our cells. So if you have a cell signaling or cell communication question on the test, there's a large chance that you're going to see the pathway. They will show you the pathway. You're going to have to just follow the path, whether it's activating, whether it's inhibiting, whether it's amplifying, or whatever is happening in the pathway. You have to just kind of follow it. And so this is one of the advantages of the paper exam, is that if it says that there's been an increase of something, you can draw a little up arrow and see, okay, well, if this is up, then that's going to be down, and this is going to be down, and that's going to be, okay. And you can follow it through. So that's what you need to make sure that you're focusing on in terms of those small, the cell communication questions.
And when they put in, again, like they're going to give all these molecules are going to have specific names, abbreviations. They're going to be like, WG4 and, interacts with this specific protein. They're just giving you labels for those molecules to give it context. Pathway still is the same.
Okay. I wrote
what
Carter.
Is cat yes?
I wrote Carter, okay? Then it corrected it and made it Darter. So I erased it and tried to write Carter again, and it wrote cat yes.
Cat yes.
So
yes,
I'm sorry, y'all. This is what happens when you try to type while you're like on a live. So the question that we have now is, can I go over G0, G1, SM, and G2? So just like a quick, what are these phases and what's happening in them?
Try to see it in your head as we go through this, just like where it is, where things exit. So G1S and G2 are going to be part of interphase, right? Named very creatively. G1, a lot of growth. S, synthesis of your DNA replication. And then G2, preparing for cell division. Guys, this is like when the cell does all the things. Like the time the cell is doing the things it's supposed to do.
90% of it.
Transcription, translation, making proteins, communicating. This is what the cell is supposed to be doing all of its life. Now, if you don't exit, my new name is Kat, yes.
Thank you, Carter.
Awesome. If you exit the cell cycle, that's going to be in the middle of G1. Basically, you've gotten cytoplasmic growth, organelle growth, and you don't go into the S phase, you're not going to copy your DNA.
It's like nerve cells, and like your liver and your muscle cells.
Yep. And then mitosis, right? So you're going to have PMAT, prophase, metaphase, anaphase, telophase, and then cytokinesis at the end. I know, listen, Georgia.
You're good.
Yeah.
Because it's totally a telophone.
You'll see telophase and you'll see cytokinesis overlapping, but cytokinesis is not a mitotic phase. It is not a stage of nuclear division. It is cellular division of the cytoplasm.
And a cell can actually go through nuclear division without going through cytoplasmic division.
We have a lot of multinucleated cells, and sometimes it's intentional, right? Skeletal muscle?
Muscle cells one, yeah. I guess it's to make extra protein. I'm not really sure. So I hope we've answered that question. We're good there. I think we're like flying through these questions. We're doing good. Ooh, let's go into evolution for a second.
HW.
Hardy Weinberg. Okay, so Hardy Weinberg. This big thing is about whether the population is going to evolve or not. So last time you did your 5 fingers, so I'm going to do my 5 fingers.
Yeah, right. We'll trade
okay?
off. Yeah.
Okay. So 5 fingers evolution. Thank you, Paul Anderson, from Boson Biology. Okay. So the 5 things I'm going to tell you is what has to be true in order for evolution not to occur. Okay. We need to have an extremely large population size. Okay. We need to have random mating. As you notice, I'm wearing my wedding band for once in my life. So we need to have random mating. You need to have no mutations, no gene flow, and then no natural selection. So nothing can be favorable or unfavorable. And so if all that is true, then theoretically the population is not going to evolve. So the way that we can tell if the population is evolving is that we can look at the frequency of the dominant allele, frequency of the recessive allele, frequency of homozygous dominant, frequency of heterozygous, and frequency of homozygous recessive. If any of those numbers change, then the population has evolved. Now, the AP exam is going to do 2 different things to you. One, they could just give you the dominant phenotype and then you calculate everything else. Or they give you the recessive phenotype, you calculate everything else, okay? Or they can tell you homozygous dominant, heterozygous, homozygous recessive, okay? There's currently a question that is the bane of my existence because I love my AP teacher friends, but they are currently not using their brain sometimes. So if they give you all 3 of those, you're going to do the allele method. You actually have to count the alleles. So take the number of homozygous dominant, multiply by 2, and then add in your heterozygous, and that will give you your frequency for your, well, divide by total alleles. So that'll give you frequency of your dominant allele. And then the same idea with the recessive. 2 times the homozygous recessive plus the 1 times the heterozygous over total. And that would give you everything. And I have a Hardy Weinberg math video. So if you go into my IGTV, you'll see that, and it will go through like the practice problems for all of the methods, and you can see exactly what I'm talking about, because I know that was a quick little 30-second boom.
And don't be afraid when you get your numbers to just like stop, like make sense of your numbers. Your numbers have to have meaning. They're not just calculations for the sake of calculation. So there's really 2 ways, and you went through them, but 2 ways to go about these problems, and very much depends upon what you're given. I'm a huge fan of starting with the homozygous recessive frequency, calculate Q if you can, but sometimes you don't even have to do that much math to get your answers. In terms of like counting the actual alleles, that, you know, not multiplying by 2 for heterozygous because they have 1 dominant and 1 recessive, that would be an unfortunate error that would happen.
Okay, so someone asked a follow-up question to that cladogram we did a second ago.
Dr. Pepsi.
Huh?
Love it. That's the username. Dr. Pepsi.
Can we just use this first row, right? Can I just use this first row here and say, okay, well, can I do everything off of E Ferris? Well, the only thing you can definitely tell without a doubt is this one. You don't know what these 2 are related to because you're seeing them related to E Ferris, so you can't do everything based on this first row. So to be very, very careful that you actually look at your entire chart when you're doing any of these problems. So that was all I wanted to add in that real quick there. Sorry.
Great. No, that makes sense.
Because I know there's a lot of students who do that. They literally just look at that first row and they try to make everything based on that. And then sometimes just stuff doesn't work out. So error bars. Are we going to see error bars?
I feel like we need a saying, like some sort of rhyme, like, you know.
For error bars?
For error bars, you know, like if they overlap, then your data's, I don't know. I need to come up with a rhyme. Anyway,
not right now. We can't do this. We have 20 minutes. We're not doing this right now.
Bars. Plus or minus 2 times the standard. I got love for my fake rap, okay? Plus or minus 2 times the standard error.
Get love for everything, child.
So if your error bars overlap at all, then there is no statistical difference between variable groups, even if their averages are different, which is why error bars give so much more oomph to your data, because they are telling you how much your data has spread from your average. So a really, really tall, wide range of data is not going to be as statistically valuable as something that's really narrow, because it's going to overlap with everything. Yeah.
Is that precision or accuracy? I always get those words.
Precision is how close you are to the average.
Okay.
No, precision is how close you are to the other groupings. That's accuracy.
And so some students have been asking me in my questions I have on my stories, like, what if they're the exact same? Like their error bars are like right at each other. Y'all, they're not trying to get you, okay? The things are going to either overlap or they
or they're
won't
not.
overlap. It's going to be obvious whether they're going to overlap or not, right? It's just,
Mm-hmm.
it's going to be obvious. You're not going to be sitting there going, oh, those
Oh,
are
do
really
they?
close. And when you do a graph, if you ever see a plus or a minus, just go ahead and say, okay, I'm going to have to put an error bar in, okay?
Mm-hmm.
Even if you don't know what you're doing, just go ahead and add it and subtract it, because look, you see all those numbers on that graph. Just
Yep.
anything you see in your table, just go ahead and put it on the graph.
Mm-hmm.
Make sure it's there. Because that's like when they first started doing that, that's what a lot of students were losing points on. And it usually, you get a point for
They
the
weren't
error
adding error
bars.
bars?
They weren't putting error bars. Like for,
Um,
I think even
Years.
now, a lot of students probably don't put error bars in their graphs. I haven't had a graphing question in like 2 or 3 years now. But they never put their error bars. And I think that they just don't know what to do with them because their teachers haven't done much with them.
Okay.
Somebody said real fast about the 5 fingers evolution. I'll say it one more time. We need to have a large population size. So that's why it's my little finger, is because you need to have a large population size. Ring finger would be, you need to have random mating. Yeah, you have random mating, so you can't be selecting for a specific characteristic, like, oh, I want that blue-footed booby because he's got the prettiest blue feet. You can't have any mutations because the mutations add new alleles, which changes your allele frequency. First finger would be no gene flow, so you can't have any immigration or emigration because that changes your allele frequency. And the no natural selection, no trait can be more favorable than another trait. So there we go. They said you have to multiply our error by 2. Whenever you're graphing, it'll actually be in there. And if it's not, the only time I've ever seen a question where it wasn't given to you, like they wanted it graphed, they had a column that said SEM, and they told you what the SEM was. And then in the prompt, they said, graph your 2 standard errors of the mean, or whatever. But that's what the SEM stands for, standard error of the mean.
Standard error of the mean.
Okay, so let's see. How about we do some chi-square real fast? So how do you do a chi-square problem? Like when you have, like if you were given a chi-square on this test, what would you do? Like, are
I
you
make a table. I'm an O minus E. I gotta like organize my thoughts because there's a lot of numbers. The math isn't hard, but it's easy to lose your way and like accidentally divide by O or something. Yeah, I make a table.
And then how do we figure out our degrees of freedom?
N minus 1. So however many options there are, subtract 1.
Okay.
Heads or tails, then there's 1 because there's 2 choices, heads or tails.
Right. And so whenever you are, after you do your chi-square, you'll get a certain value, and then you'll compare that value to the critical values that are on your formula sheet. So the formula sheet, which will be the first page of your book, or it'll be, I think, a button on the app. I haven't actually used the app, although I did find a way to get the practice test now.
Did you?
Assign it to yourself. It's crazy.
That's weird.
Yeah, no. Anyway, so you compare your number to that number. And if your number is greater than that number, then you reject the null hypothesis. If the number that you get is less than the table value, then you're going to fail to reject the null hypothesis. Yeah. I've never seen them take off for you saying except.
Oh, that's good to know. It's so awkward to remember.
They're not really that super. I've never seen them be super like over the top crazy about it. But one of the science practices I think is a chi-square. So I think that chi-square is a pretty good chance to get on there. Now, when I've looked through like practice questions, I've seen a lot of solve it, or just analyze it, or what is the degrees of freedom. So it may not be a direct like, hey, do this whole chi-square problem, or they may just ask you an expected value, like, because that's part of solving for chi-square. So repeat what I said about fail, reject, and rejecting. If the table value is less than your value, you're going to reject because your number is large, showing that there's a large amount of variance, which means that it didn't fall from the expected to the observed. And then if your number is smaller than the table value, then you will fail to reject because your number is close to what you thought, which means that we are accepting that null hypothesis.
So your variance basically means something. That's basically what you're saying.
You see what Kaufman said? Kai is high, kiss the null goodbye. Thank you, really Kaufman. Cute. You're amazing. I'm sorry she hasn't yelled at me. I don't call her by her married name. She got married like last year, and I never call her by her married name.
I think it depends. It takes a long time for that to like flush out. I still get called by my maiden name because my students are in our school for 6 to 12, and so if they know me when they're 6th graders, they will know me as that name for the next 7 years.
Okay. Oh my God, they're writing such long questions that they're so itty bitty. Can't even read these. Okay, so let's go to a replication question real fast then. So does the leading and lagging strand process occur simultaneously with the codon process? Well, so first thing, let's just talk about what's happening in
There's two
replication.
separate questions.
I know. Let's just talk about replication real fast.
Okay.
So, because I just skimmed the question, I didn't read them.
Directionality matters, everyone. DNA is anti-parallel, and use your verbs appropriately. So if we're talking about DNA replication, we are in the S phase of the cell cycle. We have DNA polymerase amongst other enzymes involved, but it's going to read the template strand 3 to 5 and synthesize 5 to 3. Right? So that's what's going to be continuous on the leading strand and discontinuous on the lagging strand because of that anti-parallel nature where my strands go in two directions. Simultaneously with the codon process, I think you're talking about the central dogma and you're asking if
I think they're asking if transcription happened at the same time as replication.
Right.
And I don't think they happen simultaneously. I think it's because it's completely different ends. Oh, everything's having different enzymes, but I think that like you can't replicate and transcribe at the same time. That's just too much going on.
The
like,
S phase
probably someone would tell me it does. But,
isn't particularly long in terms of the cell cycle, like time. I mean, it varies per cell, but I'd be surprised if there was something, if those were overlapping processes. I've not seen it. I've not heard about a question applying to that. You're going to see transcription and translation mostly during G1 and G2, not during the S phase. So I'm going to go ahead and say unlikely.
Okay. So somebody said something in the chat now, so I was trying to write back to them because they asked about like why a recessive allele is still common even though it causes problems. And they were talking about a specific answer, and I don't know the specific answers to the question that they're asking because it's very, very specific. But I did tell them to read Survival of the Sickest. So if you're interested in all that genetic stuff and why we have these genetic disorders that are harmful, like why they're still around, read that book because it's really, really cool about these genetic disorders that have been around for years and years and years because of some natural selection that happened way back in the day. So sorry. I'll get
specifically talking about polydactylism, was
do you
the
know anything
question,
about this?
Why is dominant trait polydactyl not common in human populations?
It's
very geographically accumulated polydactylism.
Brazil? I know that there's a family in Brazil that, like, because they did a whole big video thing with that family in which they play soccer and they have 6 fingers. He's a goalie. He like stops the ball. Sorry, I got to
Dominant doesn't mean more common. Recessive doesn't mean less common. I think that while there are a lot of examples in which that's true, it's not always the case.
And dominant just means that it's making something. Usually your recessive is making a non-functional protein or it's not making a protein or an enzyme. And so that's why dominant masks the recessive is because there's nothing there or it's not functional. And so that's why sometimes you could have homozygous dominant and heterozygous give you the same phenotype is because that one dominant allele makes enough that they both appear to have the correct amount of the enzyme that's needed.
Yeah. So
that was actually a question a couple years ago.
Cystic fibrosis, heterozygous advantage. Yeah. And sickle cell anemia, heterozygous advantage. And so you end up with accumulation of a recessive allele in circumstances where you wouldn't expect that to be the case. But it can happen.
So I don't know the answer to this question. I don't know if you do. That's why I'm throwing it out there. The student asked, like, what calculations do we need to know for sure? Because I guess on the probability. So definitely with probability. I would know rate. Like rate is one of the things that students always screw up on. Love you all. But somehow you hear the word rate and you just lose your minds. So definitely know how to find the slope.
And use your units.
And use your units. So there was a question, cell respiration question a while ago that students didn't get credit because they didn't use units. So naked numbers do not get you credit. I know we're not physics, but you still need numbers. I don't know. I feel like that Hardy-Weinberg is important to know.
Yeah. I don't think you're going to find yourself going to the equation sheet very often.
No.
But the conceptualization of the numbers and pulling meaning from numbers in questions, you'll have to know what the equations do and how the equations work, less so than, as many calculations as you would do in like a chem or a physics exam. Don't get bogged down in the numbers in the equation sheet. If you haven't practiced something enough to know it well, then you're gonna do your best guess and move on the exam. So, be mindful of that.
So Dr. Pepsi wants to know about the different levels of inheritance. So like what's the difference between codominance versus incomplete dominance?
So blood typing, great example of codominance with actually a recessive, right? So A and B are both codominant to the recessive O allele. And so depending upon, even though there are multiple alleles in a population, you can still only inherit two, right? You don't break the rules of fertilization with multiple allele problems. Whereas in incomplete dominance, what example do you like to give?
I like to talk about the snapdragon.
The snapdragon flowers.
Well, it's actually, snapdragons are my favorite flower.
Are they
And if
really?
you want it, yes, they really are. So if you go to a snapdragon and you squish it on the side, it goes, it's so cool. I love it. It's the coolest little flower ever. Anyways, back to my topic. So red flowers and white flowers, if you cross them together, you get pink flowers, and that's where the incomplete is, because the red allele is making red and the white allele makes white, and so red and white mixed together gives you pink. So I was like
for both these types of problems is that you get an additional phenotype. So your heterozygous produces an additional phenotype in these problems.
And so like it's also
be affected.
It's also important to know our ratios, right? So if we're looking at a monohybrid cross, you should know that a monohybrid complete dominance is a 3-to-1. You should know that a dihybrid is a 9-to-3 to 3-to-1. You should know that if we're looking at codominance with codominance with a monohybrid, it'll be a 1-to-2-to-1. And same thing with incomplete, 1-to-2-to-1. I think those are the main ones to memorize. I wouldn't really know that 6-3-3-3-2-2-1. That's just a weird one to remember. If you're wondering what that was, I think it was an incomplete dominance and a complete dominance, maybe. I can't remember what that one even was. But I know that's a ratio.
And when you express your ratios, be purposeful. Use your nouns, please.
As I just didn't. But yeah, I didn't really have a problem to go off of.
No, you didn't.
Also, make sure they're in complete sentences. If you have a free response question and they're asking you what is the phenotypic ratio, the ratio is. Give it out and have your phenotypes and everything in there. So, okay.
Ooh, we're getting down to the wire. We haven't talked about the exam much yet.
Okay, let's talk about the exam. Like, oh my God, it's 6 minutes till. Oh my God. Yeah. So we have 2 minutes. Like some people have 2 weeks left. Some people have a couple days left.
Yeah.
What do we do in this last little bit of time?
You triage. You only study the things you need reinforcement on. Let go of the things that give you the warm fuzzies because you know it backwards and forwards. You'll still know it backwards and forwards when you take the exam. So small doses, focus on the things you are weakest in, and let everything else go. Let it all go.
Yep, focus on the weaknesses. I would probably do one FRQ, one short FRQ, like one long, one short FRQ each day, just to practice. Because multiple choice questions, you can guess correctly, right? Yeah. You could just guess correctly and maybe get the points. A free response, you're not going to be able to, guess because you don't have those options there. I do those FRQ Fridays. You can easily watch some FRQ Fridays and see some of the tricks of how I say you can maybe possibly get a point if you know nothing. Right. But you're going to have to know something. And then some of the stuff you'll be able to kind of pull from the prompt. So definitely practice those FRQs. They're all on AP Central. So literally just go type in AP Bio free response, click on AP Central, and they're all there with scoring guidelines and everything, plus sample responses. So you can look over the sample response and say, okay, this student made a perfect score. What does it look like? And then Kaufman wants to remind you to look over your vocabulary. Vocabulary is how you know what the question's asking. If you don't know what the question's asking because you don't know your vocabulary, that could be a problem and why you're not as successful. And as well as that's how they can get you on the question. Not really get you, but that's how they can get you to pick an incorrect answer is because they'll use a vocabulary word in there. And since you don't know what that word means, it sounds right. You're like, yeah, pick it. So if you know your vocabulary, you're easily able to knock things out. Yeah,
it's more advantageous for you to do half the amount of problems you would want to, but understand why you got what you got on those problems. Like if you're not taking the time to reflect, learn from your mistakes, it doesn't matter if you do 200 multiple choice practice questions. If you keep doing the same things incorrectly, that's not going to allow you to have growth. So if you have a plan, do half what you can do, because you need to save half the time to reflect and look back at those choices that you may have gotten incorrect.
Wasn't it Einstein that said Sandy was doing the same thing over and over and over again,
Yeah, expecting
different responses? Yeah. So there's that. Kaufman wants to remind you not to stress about aiming for a particular score, right?
Yeah.
Just do your best. As long as you do your best, there's nothing that anyone can ask for. You did your best. You can't do better than your best. If you're needing to access the AP classroom questions, your teacher has to assign them to you. I don't think there's any way for you to get them without your teacher.
Yeah.
And I don't really want to tell you a score to aim for, because I would just want you to do your best.
Do your best.
Do your best.
Yeah, absolutely.
Whew.
Whew. This is how many lives have we done now?
This
2
is
years
number.
in a row? I know that.
Oh, us? Oh my gosh.
Yeah.
Well, we did how many did we do last year? Was it
2
3?
or 3 last year, I think.
Yeah, we did 3 last year, and then we've done each unit, so 7 because we combined some plus this. So we've done 8 lives together this year, all of which are saved on my IGTV. So if you're looking for these lives, you can go to my IGTV. You can also go to YouTube to search AP Bio Penguins, and you will pull up all of them on YouTube. You'll see all my FRQs. You actually, could go to Marco Learning, and I've done some with Marco Learning this year. You can also, type into Google the APsolute RecAP, and you could get a podcast.
You could.
In your ear, and you can walk down the street and listen. How many episodes you have for biology? Do you remember?
82. Last one comes out this Wednesday. You could binge, I think, all 5 in 12 hours, all 82 in 12 hours. So you have plenty of time. You can skip over the ones you don't need to listen to.
Yes.
But yeah.
And
so I will be hosting actually a couple of classes. So if you are looking for some more personal support, or you want to have an opportunity to ask really specific questions with visuals and everything, I'll be hosting a review class tomorrow night, as well as a class before both administrations 2 and 3. So you can find that on my Instagram. Just click on the APsolute RecAP link in my bio and sign up.
The podcast is called the APsolute RecAP, but it's APsolute RecAP. Because it's, of course, all about the AP classes. She does, what is it? You do AP Chemistry, AP Biology, AP Physics,
Music, and Gov. So we have 5 subjects and 220 episodes right now. Okay. So they're all free. You can get them on any podcast platform, Spotify, Apple Podcasts, whatever.
Well, thank you for doing this with me, Melanie. I know it was a fun undertaking that I asked you to do back in January. Like, hey, you want to do these lives with me? And you're like, yeah, let's do it. So thank you to all the students who've kind of come along with us this whole way.
Absolutely.
You are all amazing. We're proud of you for all of your hard work, for sticking it out, for doing your best. And we do hope that you come back and let us know how you did when scores come out in July, because we are very invested in you. We love you. I know. And we just want to see how well you do. And you know what? If you don't pass, please still tell us that, because you know what? There's other people who probably didn't pass too. And we can all be together and we can love on each other. And we can remember that it's been a tough year. And no matter what, that score does not define you. You're going to go to college and you will have this whole year of biology under your belt. It's going to be easier for you if you make that 3 or higher. Yes, rah, rah, woo-hoo. And you know what? We're proud of you no matter what happens. So.
And thank your teachers too. They have gone through a lot this year, and they are also super invested in your success and they want you to be, if not get a specific score, which we don't want, they want you to be proud of what you've accomplished. So please be proud. You have taken a huge undertaking. Many of you take more than 1 AP, which is admirable and impressive. These tests are not easy. They're not designed to be easy, but the resilience that you have learned through this experience is going to take you very far. I can tell you that for sure.
So with that, I'll leave you the fact that you are an AP Bio Penguin and you are dressed with success. Good night, everyone.
Have a great night, everybody. Bye.